Hi. Pancreatic cancer is often diagnosed at advanced stages, making early detection extremely challenging due to nonspecific symptoms and the absence of reliable biomarkers. In an effort to address this, we performed comprehensive serum miRNA sequencing on a substantial sample size, consisting of 212 pancreatic cancer patient samples from 14 hospitals and 213 non-cancerous healthy control samples. These samples were randomly divided into two cohorts, a training cohort, N equals 185, and a validation cohort, N equals 240. The goal was to develop and validate an accurate diagnostic model that could potentially serve as a biomarker for early pancreatic cancer detection. Using this data, we created ensemble models by combining automated machine learning with the top 100 highly expressed miRNAs. We also assessed the combination of these miRNAs with CA19-9, a known marker for pancreatic cancer. The performance of these models was then validated using the independent validation cohort. The diagnostic model that integrated the 100 highly expressed miRNAs with CA19-9 showed exceptional accuracy in distinguishing pancreatic cancer patients from healthy individuals. Specifically, the model achieved an impressive area under the curve, AUC, of 0.99, with a sensitivity of 90% and a specificity of 98%. To ensure the robustness of this model, we extended the validation process to an independent asymptomatic early stage, stage 0I, pancreatic cancer cohort. The results in this group were also promising, as the model achieved an AUC of 0.97, with a sensitivity of 67% and a specificity of 98%. This high level of diagnostic accuracy, even in early-stage pancreatic cancer cases, demonstrates the potential of these 100 miRNAs combined with CA19-9 as effective biomarkers for early detection. Early-stage diagnosis is critical in improving survival rates for pancreatic cancer patients, and our findings provide a promising avenue for improving early detection techniques. The ultimate objective of our study is to make this early detection technology available globally, primarily as a next-generation sequencing (NGS) test. However, several hurdles need to be overcome to make NGS testing feasible and sustainable. One major challenge involves the stability of miRNA profiles, which can fluctuate depending on the facility where the sample is collected. This variability can be attributed to factors like differing levels of hemolysis. Moreover, batch-to-batch differences in NGS measurements further complicate miRNA profiling, as the library preparation required for NGS measurements involves multiple reaction steps. These fluctuations in miRNA profiles can lead to decreased accuracy or even false determinations when distinguishing between cancerous and non-cancerous samples. In collaboration with ARKRAY Incorporated, our study aimed to address some of these challenges, particularly regarding the quality control of NGS measurements and sample collection. With the support of doctors from each participating facility, we achieved stable sample collection and data acquisition. However, to ensure that NGS testing becomes more widely accepted and used around the world, we recognize that improvements are still needed. Higher discrimination accuracy, reduced testing costs, and enhanced sustainability will be essential for the practical application of NGS testing for pancreatic cancer detection. To address these concerns and contribute to the advancement of NGS testing, ARKRAY Incorporated is currently developing its own in-house library preparation kit. This kit is being designed with the goal of overcoming some of the technical challenges associated with NGS measurements. In this study, we also evaluated the discrimination performance of this in-house kit in the context of pancreatic cancer detection. Validation data were collected using the in-house kit alongside the ION GeneStudio S5 system and evaluated with the pancreatic cancer discrimination model developed in our study. As summarized in the figures and table below, the results confirmed that the discrimination accuracy achieved by the in-house kit was comparable to the accuracy reported in our primary study. ARKRAY Incorporated will continue to focus on research and development, with the goal of further enhancing the performance of its in-house library preparation kit. This will ensure that the kit becomes a viable solution for NGS testing in real-world applications. The goal is to ensure that NGS testing becomes widely accessible and is integrated into society as a tool for early pancreatic cancer detection. The focus on early detection technology is crucial because pancreatic cancer is one of the deadliest cancers, and improving early diagnosis methods could significantly improve patient outcomes. The combination of advanced technologies like NGS and the development of highly specific biomarkers such as miRNAs could transform the landscape of pancreatic cancer diagnosis. By validating the performance of our models across independent cohorts and improving the robustness of the technology, we hope to make a significant impact in the early detection of this aggressive disease. Future work will involve optimizing the technical aspects of the NGS system, reducing costs, and ensuring that the testing process can be implemented on a global scale. We believe that the improvements we are making in collaboration with ARKRAY Incorporated will lead to more accurate, cost-effective, and sustainable NGS testing, which will ultimately make it possible to diagnose pancreatic cancer at much earlier stages than is currently feasible. Our study represents an important step toward this goal, but there is still much work to be done to bring this technology to widespread clinical use. As we continue to refine the technology, one of the major objectives will be to make the test more accessible and affordable, so it can be integrated into routine medical checkups, particularly for individuals at high risk of developing pancreatic cancer. This could include people with a family history of the disease or those with genetic predispositions. By offering a reliable early detection test, the overall mortality rate from pancreatic cancer could be reduced, as early-stage cancer is generally more treatable than cancer detected at later stages. In conclusion, the use of miRNAs combined with CA19-9 in NGS testing has shown significant promise in our study for early detection of pancreatic cancer. Our efforts in developing this technology, along with the in-house library preparation kit from ARKRAY Incorporated, aim to address the current challenges and limitations of NGS testing. With further development and validation, this technology has the potential to revolutionize pancreatic cancer detection and, ultimately, save lives by catching the disease in its earliest stages when treatment is most effective. We remain committed to pushing forward in this important area of research and hope to make early detection a reality for patients worldwide.